I am Ivan Duran. Today, I will talk about my research on predictive models of celiac disease onset using machine learning. Celiac disease is an autoimmune disorder. Although genetic risk and gluten exposure are necessary, they are not sufficient to trigger the onset. Recent studies show that the gut microbiome also has a role in its pathogenesis. For this research, we use data from a prospective longitudinal birth cohort study of children at high risk of celiac disease called CITIGEM, whose goal is to understand the genomic, environmental, microbiome, and metabolomic roles during early pathogenesis. In this first phase, we use cohort clinical metadata and microbiome data before onset to develop an accurate predictive tool that can tell us whether a new subject will develop celiac disease. To train the machine learning models, we use data from the first 10 cases and their matched controls by the time modeling was initiated. We did not use any data after 18 months because some subjects developed celiac disease by then. The machine learning algorithm that we use is a powerful and popular model called random forest, based on decision trees. Our data are in tables where rows are subjects and columns are different features, also called predictors. These features are either clinical metadata or the ones of microbial species, pathways, or metabolites, but not all of them are needed because they do not have predictive power and hence will worsen the model. We use an algorithm called recursive feature elimination, or RFE, which eliminated features until the desired number was achieved. First, we trained our model only using metadata. With every feature, the F1 score was 33%. By using RFE, it increased to 46% using only 4 features. This shows that clinical metadata alone are not good predictors of the onset. Next, we combine the metadata with microbiome data collected from stool samples. So we train a different model at each time point from birth until 15 months of age. We also use data from 2 time points at a time with a sliding window from birth to 15 months. Therefore, we built models for microbes, pathways, and metabolites alone. Then we integrated them with metadata, and we did the same for 2 time points. On the x-axis are the time points, and on the y-axis are the F1 scores, which go from 0 to 100. The numbers in bold and italics on the graphs are the number of features selected by RFE. The best results were by using pathways data at 9 months of age, and for 2 time points at 4 and 6 to 9 months of age, using only one pathway, which for both models was phenylalanine, tyrosine, and tryptophan biosynthesis. Next, we consider the onset to be time 0 since we knew the age when some subjects develop celiac disease. Then all time points were labeled based on how far each subject is from the onset. For instance, -6 months means 6 months before onset. So we train a different model at each time point from -18 to -3 months. We also use data from 2 time points at a time using a sliding window from -18 to -3 months. Therefore, we built models for microbes, pathways, and metabolites alone, then all of them together with metadata. The best result at 1 time point was at -6 months before onset by using only 5 features of pathways data. For 2 time points, the best result was at -15 to -12 months before onset by using only 3 features of microbial species data, which were Clostridialis bacterium, Clostridium clostridiforme, and Clostridium pathawii. In conclusion, we built highly accurate predictive models of celiac disease onset using microbiomic data both relative to the onset and absolute from birth. While clinical metadata alone are not good predictors, the best models were trained using microbes and pathways upon this data. Soon, more data from 31 cases and 62 controls will be available for us to train even more accurate predictive models. We also plan to incorporate host-derived data such as immune and cellular profiles, as well as epigenomic data. Thank you for listening, and please feel free to reach out to me or Dr. Zomorrodi at the email addresses shown on this slide.